And today we are attempting something massive. We're diving into the smallest world, but the one with the biggest impact, microbiology.
It's a huge one.
It's huge. We're going to cover the core identification, the structure, genetics, pathologies, and, the really critical diagnostic tools across all the major domains. So
Huh?
bacteriology, mycology, parasitology, and virology.
Wow.
The goal of this deep dive is really to give you a functional clinical roadmap through all of the essential microbial profiles.
It's an incredible amount of information for sure. But we're going to take our time where we need to, especially to connect the dots between, say, the structure of a microbe and the actual disease it causes, to really set the stage for just how massive this hidden world is. I want to start with a quote that it floors me every time. Neil deGrasse Tyson once said, "Within 1 linear centimeter of your lower colon, there lives and works more bacteria, about 100 billion, than all humans who have ever been born."
That quote is just stunning, isn't it? It's a perfect reminder that we're not just talking about tiny threats. We're talking about entire ecosystems living inside of you.
Entire ecosystems.
Mostly invisible, fighting these molecular wars that fundamentally determine our health and disease. So we're going to use our high-yield roadmap to break down every critical feature, starting with the absolute basics, the defining structures of bacteria.
Okay, let's do it. Let's unpack the most fundamental concept in all of bacteriology, the difference between gram-positive and gram-negative.
Right.
Because this isn't just about a color in a lab test. This is a structural difference that dictates, well, everything.
Yeah.
Which antibiotics to use, how deadly an infection might be. So why is the cell envelope the definitive feature?
Because it's basically the bacterium's identity card. The whole difference, it really just boils down to the cell wall. In gram-positive bacteria, the key thing to remember is a tremendously thick layer of peptidoglycan.
Okay.
Just think of it like a massive, spongy, protective mesh.
And that thickness is exactly why the gram stain works, right? It holds on to that first crystal violet dye.
Exactly. The thick peptidoglycan acts like a molecular trap. It just won't let go of the crystal violet. And this layer is also critically important because it's where cell wall synthesis happens.
The target for antibiotics.
The target. Any drug that disrupts that process, like penicillin, is going for the penicillin-binding proteins, or PBPs, that are right there in that wall. And unique to the gram-positives, we also find something called lipoteichoic acid, or LTA.
LTA.
It extends from the cell membrane all the way to the exterior, and our immune system sees it as a major red flag. It's highly inflammatory.
Okay, so if gram-positives have that thick trap-like wall, then the gram-negatives must have a totally different architecture that lets the crystal violet just wash right out.
Precisely. Which is why they only turn red or pink when you add the counterstain. They have a much, much thinner layer of peptidoglycan. Sometimes it's just a single layer, so the crystal violet doesn't stick at all.
But they make up for it.
Oh, they do. They compensate by adding an entire outer membrane. This extra barrier makes gram-negatives, just by design, inherently harder to penetrate with drugs.
And that outer membrane has 2 really important structures we need to focus on. First, the porins.
Yep. The porins act like selective gates or channels, controlling what gets in and out. But the component that really, really demands our attention is the notorious endotoxin,
Also known as LPS.
lipopolysaccharide. This molecule is embedded in the outer part of that outer membrane, and it is a major, major problem.
And we will get into the cascade of destruction it causes later, because it's only a problem when the cell dies and breaks apart. But that outer membrane also creates a unique little workspace, right, inside the cell wall.
It does. It creates the periplasm. This is the physical space between the inner cytoplasmic membrane and that new outer membrane, and it's exclusive to gram-negatives.
And it's a battlefield.
It's a crucial battlefield. It's where the bacteria can accumulate all their offensive tools, specifically enzymes like beta-lactamase. So when we hit the bacterium with penicillin, the beta-lactamase is sitting right there in the periplasm, ready to chew up the drug before it can even reach its target.
That's a perfect example of why the structure matters so much clinically. Okay, moving on from the envelope, let's talk about the add-ons, the structures for movement, sticking, and survival. Specifically, the ultimate survival pod, the spore.
The spore it's just nature's perfect survival mechanism. And it's important to remember it's for survival, not reproduction.
Right.
It's a dehydrated, metabolically silent structure. It's protected by this tough keratin-like coat, and inside it has its DNA dipcholic acid peptidoglycan. It's a vault. And because of this, spores can resist almost anything: dehydration, chemicals, even intense heat.
And the clinical implication there is just enormous. This is why you can't just boil your medical instruments to sterilize them.
Absolutely not. To reliably kill spores, which is essential for things like Clostridium species,
Yeah.
you have to use an autoclave. You need pressurized steam.
What's the
A
standard?
minimum of 121 degrees Celsius for 15 minutes at 15 psi. If your machine doesn't hit that spec, you haven't sterilized anything. You just made warm, wet spores.
Wow. Okay, what about the structures that help them stick to us and, spread their genes around?
So for that, you've got the flagellum, which is a long protein filament used for motility, essential for getting around. Then you have pili, or fimbriae. These are shorter, hair-like appendages.
They have two jobs.
A dual function, right? First is adherence, sticking to host cells, which is step one of any infection. And the second is for conjugation, where a special sex pilus acts like a bacterial USB cable to transfer genetic material from one cell to another.
And the protective layers outside the wall, like a coat.
Yep. Two main types. The capsule is a well-organized, discrete layer, usually polysaccharide. Its main job is protection. It's the primary shield against our professional phagocytes, like macrophages.
And the looser version.
That's the slime layer. It's more of a loose network of polysaccharides that helps with adherence, and it's the foundation of biofilm formation.
Which is how bugs stick to things like catheters and artificial joints.
Exactly. And once they form a biofilm, they are incredibly difficult to clear with antibiotics.
Okay, so if cell structure is the foundation, then the diagnostic stains are the lenses we use to actually see the threat. The Gram stain is step one, obviously, but the list of bugs that don't follow the rules, the Gram stain exceptions, that is some seriously high-yield knowledge.
I mean, honestly, knowing the exceptions is often more important than knowing the rule, because it immediately tells you something unique is going on.
And you have to know why they don't sting.
You do. So, one, they're just too thin. The spirochetes, like Treponema and Leptospira, are so skinny and delicate, they're basically invisible with a standard light microscope.
Yeah.
Two, they have a waxy wall. This is mycobacteria, the cause of TB and leprosy. They have so much mycolic acid, which is a lipid, in their cell wall that it just repels the stain.
Like water off a duck's back.
Exactly. Three, they have no cell wall at all. Mycoplasma and ureaplasma completely lack peptidoglycan, so the Gram stain literally has nothing to grab onto.
And the last group, the ones that hide.
The intracellular hiders, these are your obligate intracellular bugs, Rickettsia, Chlamydia, Legionella. They're mostly hiding inside our own cells, which makes them really hard to sample and stain effectively.
That structural knowledge, then, forces us to pull out our other tools, our specialized dyes. What are the key non-standard stains we have to know?
Okay, first up is the Agimsa stain. We use this for organisms that are either really small or live inside cells. You need this for H. pylori, Chlamydia, Borrelia, Rickettsia.
Trypanosomes, Plasmodium?
Right, it's a long list, so we use the mnemonic. Help, certain bugs really try my patience.
Ah, that's a good one. Okay, next, the targeted test for those waxy-walled bacteria, the acid-fast test.
The Deal-Nielsen stain. It uses carbolfusin, and it's the definitive test for acid-fast bacteria like Mycobacteria and Nocardia. Because it's a bit labor-intensive, a lot of labs will use the Oramine rhodamine stain for initial screening. It's fluorescent and easier to read quickly.
And finally, the quick and dirty way to see a capsule.
India ink. This is specifically for the thick polysaccharide capsule of Cryptococcus neoformans, a major cause of fungal meningitis. The ink can't penetrate the capsule, so it creates this beautiful, bright halo against a dark background. The diagnosis is basically instantaneous.
So we know their structure, we know how to see them, but getting them to actually grow in the lab, that could be the real challenge.
Yeah.
The special culture requirements in the source material really highlight how picky some of these microbes are.
Oh, absolutely. And when a bug is that finicky, its weird dietary needs become a high-yield diagnostic point in itself.
Like Haemophilus influenza.
Exactly. It's so spoiled it needs enriched media, specifically chocolate agar, because it needs two specific growth factors, factor V, which is NAD+, and factor X, which is hematin. It can't make them on its own.
And what about something like Neisseria?
For Neisseria, we often use Thiomartin agar. It's a selective medium that's loaded with antibiotics to kill off almost all the normal commensal bacteria, allowing only Neisseria to grow.
And the slow grower, TB.
Mycobacterium tuberculosis needs the specialized nutrients in Lowenstein-Jensen medium. And interestingly, that medium requires cholesterol.
Cholesterol. It makes perfect sense for a bug with such a high lipid content in its own wall.
It does. And for the GI bugs, we sort them based on the single most important metabolic trick in the gut cone. They ferment lactose.
Right, using McConkey agar.
We use differential media like McConkey agar. If the bacteria ferments lactose, like E. coli, it produces acid, and that acid turns the agar pink. If it doesn't, like Salmonella or Shigella, the colonies stay colorless. It's a simple test, but it's a massive first step in figuring out a GI infection.
And that last group, the zoonotic bugs that need that special charcoal media.
Yes, organisms like Legionella, Francisella, and Brucella, they are very fastidious and require cysteine and iron. The charcoal in the media, the buffered charcoal yeast extract, actually helps by absorbing toxic metabolic byproducts that would otherwise kill them off. It's a very specific, supportive little home for them.
Okay, let's switch gears into how these bacteria actually live, how they generate energy, how they fight back, and maybe most importantly, how they share survival information. Let's start with metabolic profiles. Oxygen seems to be the big dividing line.
It creates three absolutely critical categories. First, you have the anaerobes. They don't use oxygen, and in fact, for many of them, oxygen is toxic.
Why is that?
Because they lack the enzymes, like catalase and superoxide dismutase, that are needed to neutralize toxic oxygen radicals. So you find them in low oxygen places, mainly the GI tract and deep tissue wounds.
Clostridioids, bacteria.
Actinomyces. And clinically, you can often suspect an anaerobic infection because they tend to be really foul-smelling.
Okay, then the super flexible ones must be the facultative anaerobes.
They are the ultimate adapters. They'll use oxygen if it's around because it's more efficient for energy. But if it's gone, they can immediately switch to fermentation or anaerobic respiration. This group includes all the common players: streptococci, staphylococci, the enteric rods. It's why they can survive in so many different parts of the body.
And then there are the masters of hiding the intracellular bacteria. And we have to split those based on whether it's a choice or a requirement.
Exactly. It's a matter of dependence. The obligate intracellular bacteria like Rickettsia, Chlamydia, and Coxiella, they are total energy parasites. They have to rely on the host cell for pre-formed ATP. They literally cannot make their own.
So you can't grow them on a plate?
Never. You need a cell line. In contrast, the facultative intracellular bacteria, Salmonella, Neisseria, Listeria, Mycobacterium, they are the strategic hiders. They can live inside cells like macrophages to evade the immune system, but they have all the machinery to replicate on their own if they need to.
Okay, so now that we know where they live, let's look at the rapid lab tests we use to identify their unique metabolic fingerprints, specifically the gas they produce. Let's detail the catalase and urese tests.
The catalase test is so important. It's the key split for the gram-positive cocci. The test is simple. Can the bug break down hydrogen peroxide into water and oxygen? If it's catalase positive, you see bubbles immediately.
And that splits staph from strep.
Right. Staphylococcus is positive, streptococcus is negative. It's a super fast test with massive clinical consequences, especially for patients with chronic granulomatous disease who can't clear these catalase-positive bugs.
And urese, the ammonia producer.
Urese-positive organisms can hydrolyze urea into ammonia and CO2. The ammonia is basic, so it dramatically raises the local pH, which we see with the color change.
And this is how H. pylori survives in the stomach.
It creates its own little non-acidic bubble to live in. And Proteus, which is also urese-positive, makes the urine alkaline, which leads to the formation of those nasty struvite kidney stones. The mnemonic for these guys is Phi Chunksus.
So if the spore is their defense against the outside world, how do they defend themselves once they're inside us? Let's go back to the capsule and biofilms as their primary shields.
Right. The capsule is the anti-phagocytic shield. It's highly charged, it's hydrophilic, and it just makes it really slippery and difficult for immune cells to grab onto and eat.
And this is why the list of encapsulated bacteria, please shine my skis, is so important.
It's vital because these bugs, S. pneumonia, H. influenza, and meningitis, cause devastating infections in patients who've had their spleen removed because the spleen is absolutely essential for clearing encapsulated organisms from the blood.
And this list is also the basis for our conjugate vaccines. Can you quickly explain how a conjugate vaccine works?
Sure. The capsule on its own is just a polysaccharide. It's a T-cell independent antigen, which means it gives you a really weak, short-lived immune response. Not great.
So we conjugate it.
We chemically link, or conjugate, the polysaccharide to a carrier protein. By doing that, you engage the T cells, which creates immunologic memory, much higher levels of antibodies, and long-lasting protection. It's essential, especially for infants.
And then there are the biofilm producers. These are the ones that create a fortress on surfaces.
And it's critical to understand that a biofilm isn't just a sticky layer. It's a complete failure of our standard drug delivery model. The matrix they secrete protects the bacteria inside from both antibiotics and the host immune system.
So you have to physically remove the infected device.
Often, yes. The classic examples are S. epidermidis on catheters, P. aeruginosa in the lungs of CF patients, and Viridans streptococci causing endocarditis on damaged heart valves.
Beyond just physical shields, bacteria have these specialized protein virulence factors. These are molecular weapons designed to just dismantle our defenses. Give us the highlights.
Okay, we have to know the mechanism of these weapons. First, protein A from Staphylococcus aureus. It's brilliant.
So simple.
It binds to the SC region of IgG antibodies. So it grabs the handle of the antibody and effectively flips it around backwards. This hides the part that phagocytes are supposed to recognize, and so it completely prevents opsonization and phagocytosis.
What about IgA protease?
That's secreted by the SHine, Group S, pneumonia, H, influenza, and gonorrhea. IgA is our main antibody on mucosal surfaces. So by chewing it up, these pathogens make it way easier to colonize our respiratory or genital tracts.
And the M protein.
The M protein in strep pyogenes is another antiphagocytic factor, but its bigger, scarier role is its structural similarity to our own human cardiac tissue. This molecular mimicry is what underlies the tragic autoimmune response you see in acute rheumatic fever. The immune system gets confused and attacks the heart.
And here's where things get really frightening. Antibiotic resistance and the ability of bacteria to share this information. Let's walk through the main mechanisms of resistance first.
Resistance is complex, but it usually falls into three main buckets. 1, enzymatic inactivation. This is just destroying the drug. Think beta-lactamase chewing up penicillin.
Simple enough.
2, target modification. The bacteria changes the lock so the key doesn't fit anymore. This is how MRSA works. It alters its PBP. And it's how vancomycin-resistant enterococcus, or VRE, works. It changes the cell wall precursor so vancomycin can't bind.
And the third way.
Efflux, or decreased uptake. So either pumping the drug right back out of the cell as fast as it comes in, or closing the door so it can't get in to begin with.
But the real crisis is that these resistance blueprints don't stay in one bacterial family. They get shared horizontally. How do they do that?
This horizontal gene transfer is what's driving the entire global resistance crisis. There are 4 key mechanisms. First is transformation.
Okay.
This is just the uptake of naked DNA from the environment. A bacterial cell dies and lyses, its DNA spills out, and another competent cell can just come along and pick it up. This is a common mechanism for S. pneumonia and H. influenza.
Second is conjugation, the bacterial USB cable.
Right. Conjugation is a direct, programmed cell-to-cell transfer of a plasmid through a sex pelus. The donor cell, which is $5 pluser, gives a copy of its F plasmid to a $5 recipient, turning it into a $5 plus cell.
And the high-stakes version of this, HFR, is when that F plasmid gets integrated into the chromosome.
Correct. In high-frequency recombination, or HFR, the integrated plasmid tries to transfer itself, but because it's now part of the chromosome, it starts pulling huge chunks of the host's chromosomal DNA along with it, transferring all sorts of new genes.
Okay, third mechanism, transduction. This one involves viruses.
Yes. Transduction uses bacteriophages viruses that infect bacteria as the delivery vehicle. You have generalized transduction, which is basically a packaging mistake. A random piece of bacterial DNA gets accidentally stuffed into a new phage head.
But specialized transduction is the really scary one.
It's far more specific and honestly more chilling. This involves lysogenic phages that integrate their DNA into the bacterial genome. When the phage decides to leave, it sometimes makes a mistake when excising itself and grabs the adjacent bacterial genes along with it.
Wait, so a virus is the delivery truck for lethality?
That's exactly what it is. And this is one of the most important facts in all of microbiology. The virulence genes for the ABCDE group of toxins are not on the bacterial chromosome. They are encoded on the phage DNA.
So a harmless bacteria can become a killer just by getting infected by the right virus.
Precisely. The list includes the toxins for A, strep erythrogenic, botulinum, cholera, diphtheria, and E, EAAC's Shiga toxin.
Wow. And the fourth mechanism?
Transposition. This is via transposons or jumping genes. These are mobile segments of DNA that can cut themselves out and paste themselves into a new location on either a plasmid or the chromosome. This is how the Van-A resistance gene for vancomycin can jump from, say, an enterococcus to S. aureus. It's the ultimate mechanism for rapid, widespread distribution of resistance.
Okay, now let's detail the actual weapons. We set up the contrast before. Exotoxins versus endotoxins. If exotoxins are the engineered surgical strikes, let's start with the catastrophic collateral damage caused by that structural component, the endotoxin.
That structural component, lipid A of the LPS, is everything for endotoxin. Remember, exotoxins are proteins secreted by both gram-positives and gram-negatives. They're highly specific, high-toxicity weapons.
Whereas endotoxins are part of the building.
Exactly. They are structural components exclusive to the outer membrane of gram-negative bacteria, and they're only released when the cell lyses and falls apart.
Let's break down that comparison table from the source, focusing on the danger.
Look at the difference in stability. Exotoxins are incredibly fatal in tiny, tiny doses, but they're heat labile. They denature pretty easily, around 60 degrees Celsius. Endotoxins, on the other hand, are less fatal per dose, but they are incredibly heat stable.
How stable?
They can survive being boiled at 100 degrees Celsius for an hour. And that heat stability is a public health nightmare. You can autoclave your surgical instruments to kill all the bacteria. But if gram-negative bacteria were there, at least the endotoxin is still there, active, ready to cause a fever. It's a pyrogen.
And what about antigenicity? Can we make vaccines against them?
For exotoxins, yes. Because they're proteins, they are highly antigenic. They provoke a strong immune response, and we exploit that to make toxoid vaccines, like for tetanus and diphtheria. Endotoxin, however, is poorly antigenic, so no. There is no protective vaccine against gram-negative endotoxic shock.
Okay, let's detail that pathway. The destructive cascade of endotoxin shock, because it leads to systemic failure so quickly.
When that lipid A is released, it triggers 3 parallel catastrophic events at once. 1. Macrophage activation. This is the main driver. Macrophages just dump inflammatory cytokines into the system. TNF-alpha, which causes fever, shock, and vasodilation. IL-1, more fever, and IL-6. This is the cytokine storm.
Okay, what's second?
Complement activation. This releases C3A and C5A, which further trigger histamine release, more vasodilation, more leaky blood vessels.
And third, this is where the clotting system breaks down.
Third is tissue factor activation. This is where the patient's entire coagulation cascade gets turned on, and it can lead directly to disseminated intravascular coagulation, or DIC.
Wait, DIC from just this one part of a dead bacterium cell wall? That is staggering.
It is. DIC is this catastrophic failure where the body is both clotting everywhere, creating microthrombi that block organs, and bleeding uncontrollably because all the clotting factors have been used up.
And it happens fast.
Incredibly fast. You go from a localized infection to septic shock to multi-organ failure because that one single molecule, lipid A, is an indiscriminate on switch for systemic inflammation and clotting failure. The bacteria doesn't even have to be alive.
Wow. Okay, now for the surgical precision of the exotoxins. Let's start with the ones that inhibit protein synthesis.
The theme here is stopping the cell's factory. They shut down its ability to make essential proteins, often through a process called ADP ribosylation.
Like diphtheria toxin.
Diphtheria toxin and P. aeruginosa's exotoxin. A, both target and inactivate a key protein called elongation factor 2, or EF2. This stops protein synthesis dead in its tracks, leading to cell death and the destructive local symptoms, like that pseudomembrane in diphtheria.
And Shiga toxin works differently.
It achieves the same goal, but it does it by inactivating the 60S ribosome itself. In EAG, the Shiga toxin has a special affinity for the capillaries in the kidney, which is what leads to hemolytic uremic syndrome, or HUS, that triad of mechanical hemolysis, low platelets, and acute kidney failure.
Okay, next up, the toxins that cause massive fluid loss. The CAMP and CGMP activators.
These toxins hack the signaling pathways in our intestinal cells, forcing them to just pump out chloride and water.
The ETEC toxins.
Right. The heat labile, LT toxin, overactivates adenylyl cyclase, which increases CAMPP. LT is labile, so it's stable in the air, rising CAMP. The heat stable ST toxin overactivates guanylate cyclase, increasing CGMP. ST is stable, so it's stable in the ground.
And cholera toxin works like LT.
Almost identically. It permanently activates adenylyl cyclase, leading to that massive, unending rice water diarrhea.
And now the most terrifying ones? The clostridial neurotoxins that cleave snare proteins. It's fascinating that two toxins with almost the exact same molecular mechanism can have clinically opposite outcomes.
It's all about the target neuron. Both tetanospasmin from C. tetani and botulinum toxin from C. botulinum are proteases that cleave the snare complex.
And that prevents neurotransmitter release.
Right, it prevents the vesicles from fusing with the membrane. But tetanospasmin travels up to the central nervous system and blocks the release of inhibitory neurotransmitters, GABA and glycine.
So there's no stop signal.
No inhibition. The motor neurons fire constantly, and you get spastic paralysis, lockjaw, the arched back, opisthoton.
With botulinum toxin.
It acts locally at the neuromuscular junction, and it blocks the release of the excitatory neurotransmitter, acetylcholine.
So there's no go signal.
And you get flaccid paralysis.
Yeah.
Descending weakness, floppy baby syndrome. One prevents the stop signal, the other prevents the go signal. It's a stunning example of molecular specificity.
Absolutely. And then you have the tissue degraders.
Like the alpha toxin from C. perfringens, which is a phospholipase that just liquefies cell membranes and tissue, causing gas gangrene. And streptolysin O from S. pyogenes, which is a hemolysin we can actually measure to diagnose a recent strep infection.
Finally, superantigens, the nonspecific immune bomb.
Superantigens from S. aureus and S. pyogenes cause massive damage by just bypassing the normal specific antigen presentation process. They essentially force the immune system to fire every gun it has all at once, indiscriminately.
How do they do that?
They physically cross-link the T-cell receptor directly to the MHC class II molecule on an antigen-presenting cell. This leads to a massive, non-specific release of cytokines, IL-2, interferon gamma, and tons of TNF-alpha, which culminates in the fever, rash, and multi-organ failure you see in toxic shock syndrome.
All right, now we get to use that lab algorithm to apply all the structural and metabolic knowledge to the specific players, starting with the gram-positive organisms. So after confirming they're gram-positive, the first split for the coccis, the spheres, is the catalase test.
Absolutely. Catalase negative, it's a streptococcus. Catalase positive, it's a staphylococcus. And within staphylococcus, the next critical test is coagulase. That splits the field wide open.
And coagulase positive is almost always S. aureus. That's the gold standard for identifying the most virulent member of this family.
For sure. S. aureus virulence is complex. It relies on that protein A we talked about to block phagocytosis and coagulase to clot fibrinogen, which helps it wall off abscesses. It causes 3 major types of disease: inflammatory, like skin infections and osteomyelitis, and the really scary, rapid, toxin-mediated diseases.
Toxic shock, scalded skin syndrome, food poisoning.
Right. And of course, the greatest clinical challenge globally, MRSA.
Methicillin-resistant S. aureus.
This is target modification at its absolute worst. The resistance is from the MECA gene, which codes for a modified PBP. The beta-lactam antibiotics just can't bind to it effectively anymore, rendering them useless.
And the coagulase-negative staph are split by the novobiosin test.
Right. S. epidermidis is usually normal skin flora, but it's a huge cause of prosthetic device infections because of its amazing ability to form biofilms. It's novobiosin sensitive. S. saprophyticus, on the other hand, is novobiosin resistant, and it's the number 2 cause of uncomplicated UTIs in young, sexually active women.
Okay, moving to the catalase-negative group, streptococcus. Here we use hemolysis patterns on blood agar as the sorting mechanism.
Exactly. We're looking for one of 3 things. Alpha hemolysis, which is partial, giving a greenish color. Beta hemolysis, which is complete lysis, a clear zone. And gamma hemolysis, which is no hemolysis at all.
Let's focus on the alpha hemolytic guys and the 2 quick tests that split them.
So one is Streptococcus pneumonia. It's encapsulated, looks like lancet-shaped diplocacci. It is sensitive to a chemical disc called optochin, and it's bile-soluble. It's the leading cause of MOPS, meningitis, otitis media, pneumonia, and sinusitis.
And the other alpha.
The viridins group, Streptococci. They are optochin and bile-resistant or insoluble. They're part of our normal oral flora, but they're notorious for causing infective endocarditis. One of them, S. sanguis, makes dextrans, which is like a glue that lets it stick firmly to already damaged arc valves.
Now for the beta hemolytic group, the complete destroyers.
2 big ones here. First, Streptococcus pyogenes, or Group A strep. It's bacitracin sensitive. The key is to remember it's 3 types of disease: pyogenic, like strep throat, toxinogenic, like scarlet fever, and immunologic.
And the immunologic is the most feared.
Absolutely. Rheumatic fever and post-strep glomerulonephritis. And remember, that M protein is the antigen that fools our immune system into attacking our own heart and kidneys.
And the other beta hemolytic. Group B.
Streptococcus agalactio, or Group B strep. It's bacitracin-resistant and CAMP positive. And you remember Group B for babies. It's the leading cause of neonatal sepsis and meningitis. This is why all pregnant women have to be screened for it late in pregnancy and get antibiotics during delivery if they're positive.
Got it. And briefly, the gamma hemolytic bugs, Enterococcus.
Enterococcus is tough. It can grow in high salt and bile, it causes UTIs, and clinically the biggest concern is the rise of VRE, vancomycin-resistant enterococci, in hospitals.
Okay, let's quickly hit the key gram-positive rods, or bacilli, which often rely on spores and unique toxins.
Right, 1. Bacillus anthracis, the classic spore-forming rod. Its lethality comes from its 2 toxins, edema factor, which increases CAMP and causes swelling, and lethal factor, which cleaves a MAP kinase and causes necrosis. Pulmonary anthrax is incredibly lethal.
Number 2.
2. Clostridioides difficile, the ultimate side effect of modern medicine. It's a spore former, an obligate anaerobe. Its toxin A and toxin B cause diarrhea and pseudomembranous colitis, almost always after a course of antibiotics has wiped out the normal gut flora.
And 3. Listeria monocytogenes.
A facultative intracellular rod we get from contaminated food. Its unique survival trick is actin polymerization. It hijacks the host cell's own actin to build a little rocket tail, which propels it from one cell directly into the next.
So it avoids antibodies completely.
Completely. And it's a major cause of meningitis in newborns, the elderly, and the immunocompromised.
Finally, for the gram-positives, the branching filaments that often look like fungi. Nocardia versus actinomyces.
This is a classic differential. Nocardia is aerobic, catalase positive, and weakly acid fast. It's found in the soil, causes lung infections that look like TB, and often spreads to the brain. Treatment is sulfonamides.
And actinomyces.
The opposite in every way. Anaerobic, catalase negative, and not acid fast. It's part of our normal oral and GI flora. It causes these draining abscesses and sinus tracts that contain characteristic sulfur granules. Treatment is penicillin.
And the most important acid-fast organisms, mycobacteria.
Slow-growing, acid-fast rods. The big one is M. tuberculosis. Primary infection can result in a quiet, calcified Gone complex. The real disease is secondary or reactivation TB, which gives you those cavitary lesions in the upper lobes of the lungs.
And the immune response involves those Langerhans giant cells.
Exactly. Fused macrophages trying to wall off the infection. And clinically, reactivation is a massive risk for anyone on immunosuppressants, especially TNF-alpha inhibitors.
Okay, switching sides to the gram-negatives, which stain red or pink. Let's start with the only medically significant gram-negative kochi, the Neisseria species.
Right. Neisseria are diplicaci. They both produce IgA protease and are often found hiding inside neutrophils. The differentiation between them is crucial.
Gonokochi versus meningokochi.
Exactly. Gonokochi, N. gonorrhea, has no capsule and does not ferment maltose. It causes gonorrhea and is the most common cause of septic arthritis in young adults.
And meningokochi.
N. meningitatis has a polysaccharide capsule and does ferment maltose. It's transmitted by respiratory droplets and causes severe meningococemia, which can lead to rapid adrenal hemorrhage and death, a condition called Waterhouse-Friderichsen syndrome.
Okay, next, the gram-negative rods, starting with the ones that hit the respiratory tract.
First up, Haemophilus influenza. It needs that chocolate agar. The non-encapsulated strains cause common stuff like ear infections, but the encapsulated type B strain causes severe disease meningitis and, classically, epiglottitis.
The cherry red epiglottis.
Right, which can cause total airway obstruction. The high B vaccine, which is a conjugate vaccine, targets that type B capsule.
And the whooping cough bug.
Bordetella pertussis. Its pertussis toxin increases KMP, which destroys the ciliated cells in our respiratory tract. This leads to those characteristic violent coughing fits, followed by that high-pitched inspiratory whoop. Prevention is the Tdap vaccine.
All right, let's tackle the massive group of enteric rods, which we already sorted using McConkey agar. Lactose fermentation is the key divider.
It is. The lactose fermenters are the Citrobacter, Citrobacter, E. coli, Enterobacter, Klebsiella, Serratia.
And for E. coli, we have to know the different strains.
For sure. E-Tech causes traveler's diarrhea with its LT and ST toxins, but EHEC, specifically O157H7, is the dangerous one because its Shiga toxin can lead to hemolytic uremic syndrome, HUS.
And Klebsiella.
Highly virulent because of its massive mucoid capsule. It's famous for causing aspiration pneumonia, especially in alcoholics, leading to that classic currant jelly sputum.
On the other side of the plate, the non-lactose fermenters, which are mainly Salmonella and Shiga, we need to meticulously break down their differences.
This comparison is extremely high yield. So first, reservoir. Salmonella is found in humans and animals. Shigella is humans only.
Right.
Motility and H2S production. Salmonella is motile and produces H2S, which turns the colonies black on certain agar. Shigella is non-motile and does not produce H2S.
And the infectious dose? This is a big one.
A huge one. For Salmonella, you need a high dose because it's acid labile. It gets killed by stomach acid. For Shigella, the infectious dose is incredibly low because it's acid stable. Just a handful of organisms can cause disease.
Which is why it spreads so easily from person to person.
Exactly. And finally, the pathology. Salmonella causes diarrhea, or in the case of S. typhi, typhoid fever. Shigella causes dysentery, a really bloody mucoid stool.
Now for the curved rods, which cause major GI problems.
First, Vibrio cholerae. Comma-shaped, oxidase positive, it produces the cholera toxin leading to that voluminous non-inflammatory rice water diarrhea.
And the ulcer bug.
Helicobacter pylori. Curved, flagellated, and strongly urese positive. It uses that urese to survive in the stomach, where it causes gastritis, ulcers, and increases the risk of gastric cancer. Treatment is that complex triple therapy with 2 antibiotics and a PPI.
Finally, the elegant spiral-shaped bacteria, the Spiracetes, too thin for gram stain, moving with those internal axial filaments.
The 3 big ones are Leptospira, Treponema, and Borrelia. Let's focus on the last 2. Lyme disease is caused by Borrelia burgdorferi. It's transmitted by the Ixodes tick, the same one that carries Babesia.
And it has 3 stages.
Stage 1 is the localized erythema migrans, the classic bullseye rash. Stage 2 is disseminated with neurologic problems like facial nerve palsy and cardiac block. And stage 3 is chronic arthritis and neuropathy.
And syphilis, caused by Treponema pallidum.
Also has stages. Primary is the painless chancre. Secondary is the disseminated disease with a systemic rash that uniquely involves the palms and soles, and these lesions, called condylomata lata.
And tertiary.
Chronic destruction, gummas, aortitis, and neurosyphilis. And for diagnosis, you start with a non-specific screening test like VDRL or RPR, and then you confirm with a specific test like FTA-ABS.
We're moving now into mycology. Fungi. These are eukaryotes. They're more complex than bacteria, and their infections are so often tied to geography and a person's immune status. The first big structural principle we need is the dimorphism rule.
It's a simple and powerful rule. Mold in the cold, yeast in the heat. So the systemic mycoses exist as these fuzzy moles out in the environment at 20 degrees Celsius. But when they get inside a warm human host at 37 degrees, they convert to the invasive single-celled yeast form.
But there's an exception.
Of course. The major exception is coccidioids. It's a mold in the cold, but in the heat, it forms a large, non-budding spherule that's filled with endospores.
Okay, so let's link the key systemic mycoses to their geography and their unique pathology. Knowing the environmental source is often half the diagnosis.
For sure. So histoplasmosis. Think Mississippi and Ohio River valleys. It's associated with bird or bat droppings. Pathologically, you see macrophages stuffed with tiny histoyeasts smaller than a red blood cell.
Okay.
Coccidioidomycosis, southwestern U.S., California. Associated with desert soil and dust, causing desert rheumatism. Pathologically, you see that spherule filled with endospores.
And the third one?
Paracoccidioidomycosis, Latin America. And its pathological feature is a budding yeast with this classic captain's wheel formation.
Moving to the opportunistic fungal infections, the ones that attack the immunocompromised.
Candida albicans is number 1. It's dimorphic, but it's unique because it forms these things called germ tubes when you culture it at 37 degrees. It causes everything from thrush and vaginitis to life-threatening systemic candidemia.
And Aspergillus?
Aspergillus fumigatus. You identify it by its sharp, acute angle, 45-degree branching septate hyphae. It can cause allergic disease, fungal balls called aspergillomas, or invasive disease. Critically, some strains produce aflatoxins, which are strongly linked to liver cancer.
And finally, Cryptococcus.
Cryptococcus neoformans. This is the heavily encapsulated yeast found in pigeon droppings. It's the leading cause of fungal meningitis in AIDS patients, and we diagnose it with that India ink stain or by testing for the cryptococcal antigen.
Now for parasitology. Let's start with the protozoa that cause GI and CNS infections.
Okay, for GI protozoa, Giardia lamblia causes fatty, foul-smelling diarrhea because it messes up fat absorption. You get it from cysts in contaminated water.
The classic camper's diarrhea.
That's the one. Entamoeba histolytica causes a bloody diarrhea and can form huge liver abscesses that are filled with this anchovy paste exudate. And cryptosporidium causes a severe, persistent watery diarrhea, especially in AIDS patients.
And the CNS protozoa are terrifying. They really show how parasites can exploit our anatomy.
They do. Toxoplasma gondii is acquired from cysts in undercooked meat or oocysts in cat feces. In an AIDS patient with a CD4 count under 100, it causes these classic multi-ring enhancing lesions on an MRI, which are brain abscesses.
And the brain-eating amoeba.
Naegleria fowleri. It's found in warm, fresh water. It enters the CNS by traveling up the olfactory nerve, straight through the cribriform plate in the skull, and causes a rapidly fatal meningoencephalitis.
Now for the vector-borne protozoa that cause blood and visceral infections.
Malaria, caused by Plasmodium, is transmitted by the Anopheles mosquito. Different species cause different cyclical fever patterns. Diagnosis is by looking for the classic trophozoate ring forms inside red blood cells on a peripheral blood smear.
And babesiosis.
Babesiosis, caused by Babesia, is transmitted by the ixodes tick, the same vector as Lyme disease. It causes fever and hemolytic anemia, and its unique diagnostic sign is the Maltese cross formation on a blood smear.
And Chagas disease.
Caused by Trypanosoma cruzi, transmitted by the reduvid kissing bug. The chronic disease is characterized by the destruction of nerves in the heart and GI tract, leading to dilated cardiomyopathy, megacolon, and megaesophagus.
Let's run through the helminths, the worms, but let's focus on the transmission route for the nematodes and the key clinical points for the cestodes.
Sounds good. For nematodes, the roundworms, it's all about how you get them. Some are ingested. Enterobius, pinworm, ascaris, giant roundworm. Others, the larvae, penetrate your skin from contaminated soil. The hookworms, ancylostoma and nectator, which cause anemia by sucking blood, and strongyloids.
And the cestodes, the tapeworms, where the mode of ingestion completely changes the disease.
This is critical. For Taenia solium, the pig tapeworm, if you eat the larvae in undercooked pork, you just get an intestinal tapeworm. But if you accidentally ingest the eggs from food contaminated with human feces, the eggs hatch and the larvae migrate all over your body, causing cysticercosis with cysts in your brain and muscle.
A massive difference.
Huge. And Diphyllobothrium laudum, the fish tapeworm, is the longest worm. It loves vitamin B12 and competes with you for it, which can lead to a megaloblastic anemia.
And the flukes, or trematodes?
The most important is cystosoma. The intermediate host is a snail, and the infectious larvae penetrate your skin in contaminated water. S. hematobium specifically loves the bladder, causing chronic hematuria and increasing the risk of bladder cancer.
Okay, we've covered the cellular threats. Now we shift to the ultimate simple infectious agents, viruses. Just nucleic acid and a protein coat. But that simplicity leads to incredible complexity in replication and, more importantly, mutation. Let's start with viral structure and genetics.
Viruses are just genetic material DNA or RNA inside a capsid, which is a protein coat. If they have an envelope, it's a lipid bilayer they stole from the host cell membrane.
That makes a big difference.
A huge difference. Enveloped viruses are fragile. They're easy to kill with disinfectants. Non-enveloped or naked viruses are much tougher and can generally survive the harsh environment of the GI tract.
The real trickery with viruses, though, is how they can swap parts. Let's distinguish between the two main ways they exchange genetic material, starting with the one that causes major pandemics.
That would be reassortment. This is the mechanism for viruses that have segmented genomes like influenza A and rotavirus. If two different strains of flu infect the same cell, they can just swap entire segments of their genome. This leads to a dramatic, rapid change in their surface antigens.
Which is known as antigenic shift.
Exactly. And that shift is why we get major pandemics and why the flu vaccine has to be reformulated every single year.
And the other, more common exchange.
That's recombination, which is just crossing over between two homologous sequences. And then there's phenotypic mixing, which is just a transient change. A new virus gets packaged with the coat proteins of a co-infecting virus, but its own genome is still inside, so the next generation reverts back to normal.
Okay, if structure is king, then the DNA and RNA rules are where we start. But as always, there are exceptions.
Always. So for DNA viruses, the rule is they are all double-stranded except for parvovirus, which is single-stranded. And all DNA viruses replicate in the nucleus except for pox virus. Pox is out of the box. It replicates entirely in the cytoplasm.
And for RNA viruses?
For RNA viruses, the rule is they are all single-stranded except for Reoviridae, which is the family that includes rotavirus. It's double-stranded and segmented.
Let's focus on the largest and most clinically diverse DNA family, the herpes viruses. And their defining feature is latency.
They are all enveloped double-stranded linear DNA viruses, and where they hide, their latency site is critical.
Okay.
HSV-1 hides in the trigeminal ganglia. It causes cold sores and is the most common cause of fatal temporal lobe encephalitis.
VZV.
The VZV hides in the dorsal root ganglia. Primary infection is chickenpox. Reactivation is shingles.
EBV.
EBV hides in B cells. It causes mono, the kissing disease, and is linked to Burkitt lymphoma. And CMV hides in mononuclear cells. It causes severe disease in newborns and transplant patients, often with those classic owl eye intranuclear inclusions. And don't forget HHV-8, which causes Kaposi sarcoma.
Okay, moving to the vast world of RNA viruses. Let's start with the small naked ones.
The picornaviruses. They're non-enveloped, single-stranded RNA viruses. Think perch poliovirus, echovirus, rhinovirus, Coxsackie, HAV. Critically, rhinovirus, the common cold, is acid labile. It gets destroyed by stomach acid, so it can't cause GI disease. Polio and HAV, on the other hand, are acid stable.
And the heavy hitters, the retroviruses, with their unique replication strategy.
Retroviruses, like HIV, are enveloped. They use an enzyme called reverse transcriptase to make a double-stranded DNA copy of their RNA genome. And that DNA copy then integrates permanently into the host cell's DNA.
Which is why it's a lifelong infection.
Exactly. That integration is what makes HIV so hard to cure and requires lifelong therapy.
And influenza, the master of antigenic shift.
Orthomyxoviruses.
Yeah.
Enveloped and segmented, which is what allows for that reassortment and antigenic shift, driving the constant need for new vaccines.
Okay, let's dedicate some serious time to the hepatitis viruses. Understanding their differences in structure and their risk of becoming chronic is paramount.
For sure. There are 5 major types. The key distinction is transmission and chronicity.
Let's run them down.
Hepatitis A, HAV, is a picornavirus. Fecal oral transmission. It's acute only. No chronic risk. Hepatitis B, HBV, is a hepatinovirus, a DNA virus, transmitted parenterally or sexually. Big risk of chronic disease and cancer. Hepatitis C, HCV, is a flavivirus, transmitted parenterally. This is the major blood-borne chronic threat. Also a big risk for cancer. Hepatitis D, HDV, is a defective virus. It needs HBV's envelope to survive, so you only get it if you also have Hep B. And hepatitis E, HEV, is fecal oral like A, but it's especially dangerous for pregnant women. No chronic risk.
That is fascinating. HBV, a DNA virus, uses reverse transcriptase to replicate, which is why we can treat it with drugs developed for retroviruses.
It's a very strange hybrid mechanism, but the most important and often the most confusing high-yield concept is the hepatitis B serology. We need to walk through this slowly. We monitor 4 main markers.
Okay, let's establish the baseline. What tells you someone is currently infected?
That would be HBsAg, the surface antigen. If that's present, the patient is infectious either acutely or chronically.
And what tells you they're protected?
Anti-HB is the surface antibody. This means one of 2 things. Either they recovered from a natural infection or they were immunized. It's the antibody of protection.
So if I was successfully vaccinated against hepatitis B, what should my panel show?
It should show only anti-HBs and nothing else, because you were never actually exposed to the core of the virus, just the surface protein.
Okay, so what about that core antibody, anti-HBC?
Anti-HBC means you were exposed to the core of the virus, which only happens during a natural infection. The IgM subtype, anti-HBC IgM, is the definitive marker of an acute or recent infection. And this is critical for diagnosing someone in the window period.
Ah, the notorious window period. This is when the HB-Sag has been cleared, but the protective anti-HCPs hasn't shown up yet. So how do we spot an infection then?
The HBsAg is gone. The anti-HBs is still too low to detect. The only marker present during that 1 to 2 month gap is anti-HBC IgM. It's the only thing that tells you there was a recent infection. It bypasses all the ambiguity of the window.
Okay, we conclude our virology deep dive with HIV, the retrovirus that basically generates immune suppression.
Right. HIV uses its surface proteins, GP120 and GP41, to bind to CD4 and the CCR5 or CXCR4 co-receptors on T cells and macrophages. The standard diagnosis now tests for both HIV antibodies and the P24 antigen, which is a core protein that shows up early.
And the definition of AIDS is critical. A CD4 count below 200, or the presence of an AIDS-defining condition. The clinical map of which infections show up, at which CD4 level is a chilling illustration of progressive immune failure.
It really connects the dots. So at a CD4 count below 500, you start to see things like oral thrush, candida, oral hairy leukoplakia, EBV, and reactivation of TB.
And below 200.
CD4 below 200 is the formal AIDS threshold. The key infections here are pneumocystis jirovecii pneumonia, or PCP, which shows up as ground glass opacities on imaging, and disseminated histoplasma. This is where prophylaxis becomes mandatory.
And below 100 is severe.
CD4 below 100 is severe immunodeficiency. This is the CNS zone. You see Cryptococcus meningitis, CMV retinitis, and toxoplasma brain abscesses. The lower the count, the more aggressive and unusual the organisms that can gain a foothold.
We have just completed a full sprint through the microbial world. We've tackled the identity, the weapons, the defenses of everything from the smallest spiricate to the most complex retrovirus. We have gone line by line through the structure of a spore, the mechanism of a superantigen, and the crucial serology of hepatitis B. You now have, I hope, an incredibly structured mental map of these microbial threats and the concepts we use to fight them.
And this knowledge is so far from just being academic. I mean, understanding these molecular mechanisms is the key to global defense. The concepts we covered today, like the way the mecha gene modifies a target to cause MRSA or the process of horizontal gene transfer, these are the engines driving the next public health crisis.
These tiny biological events have massive consequences.
Absolutely.
Which brings us to our final provocative thought for you, the listener. We spent some time detailing specialized transduction, that bizarre process where a relatively benign bacterium can acquire its killer genes, like diphtheria or cholera toxin, from a bacteriophage, basically a virus that infected it.
So consider this. If a virus is responsible for making a non-virulent bacterium deadly, what does that really imply about the future of microbial evolution and our ability to control it?
What happens when these highly mobile genetic elements, these viral delivery trucks and jumping transposons, jump to entirely new bacterial hosts that we aren't even watching yet?
The battle against infection is never static. It is defined by the constant, rapid movement of lethality between species. Go forth and keep learning.
We'll see you next time for the next Deep Dive.